Welcome back to your channel. Today we will discuss important definitions in pharmaceutical industry. Whether you are a seasoned professional or just starting out, understanding these definitions is crucial for ensuring that medications are safe, effective and produce consistently. In this video we will explore these definitions. So if you are ready to enhance your knowledge about how the pharmaceutical industry maintains high standards and ensures patient safety, hit the subscribe button and let's get started. Quality. Origin of the word quality. The term quality is derived from the Latin word qualitas, which means what kind. This origin hints at the nature or type of something, emphasizing its essential characteristic. Definition in the context of medicinal products. In pharmaceutical industry, quality refers to the totality of features and characteristic of medicinal product. This means it encompasses all aspects that define the product standard, including purity, efficacy, safety, and stability. Quality is measured by a product's ability to satisfy stated or implied needs. Stated needs are those explicitly outlined like regulatory standards, while implied needs are those expected by users such as consistent performance and safety. Therefore, quality is a comprehensive concept. In pharmaceuticals, it represents a product's complete ability to fulfill both regulatory and user expectations, ensuring it is safe, effective, and reliable. Quality assurance definition. Quality assurance or QA is defined as the totality of arrangements put in place to ensure that pharmaceutical products meet the necessary quality standards for their intended use. This means quality assurance involves all the planning, processes, and protocols aimed at maintaining and improving the quality of these products. QA is a comprehensive approach that covers all aspects affecting quality either individually or collectively. This includes various areas like raw material selection, manufacturing process, testing, and storage conditions, all of which contribute to final product quality. And the objective of quality assurance, the primary objective of quality assurance is to ensure that each product not only meets regulatory requirements but also consistently perform as expected by both regulators and users. It establishes a robust system that minimizes risks and guarantees product safety and efficacy. Overall, quality assurance is about creating a reliable framework to deliver high quality pharmaceutical products. By covering all the factors impacting quality, it assures patients and healthcare providers of the product's suitability and effectiveness for its intended therapeutic purposes. Quality control. Quality control refers to a procedure or series of steps applied during the manufacturing process to ensure that the product or service consistently meets required standards. This means QC is focused on verifying that every product batch adheres to a quality specifications and is reproducible in the terms of quality. QC is a vital part of good manufacturing practices, particularly in pharmaceutical industry. It involves specific activities like sampling, testing, documentation, and release procedures. These steps ensure that each batch undergoes the necessary tests to verify its quality before it reaches the market. QC includes a detailed and systematic process where products are sampled and tested to confirm they meet established quality criteria. Documentation ensures that all results are recorded and traceable, and only when the product passes all the quality checks, it is approved and released for the customer use. Standard. A standard is something established as a benchmark or model, which other similar items or materials are expected to match. In the context of pharmaceuticals, a standard ensures that products meet consistent quality, potency, and safety levels across the different batches. Standards are categorized into two different main types, reference standard and working standard. Reference standard is the highest quality benchmark, often a pure substance or material with established quality parameters. It serves as a definitive measure for testing and verifying the quality of other samples. Working standard. Derived from the reference standard, the working standard is used routinely in the laboratories for daily quality control testing. It is calibrated against the reference standard to ensure accurate and reliable testing results. What is reference standard? A reference standard is a drug substance with the highest possible purity prepared through independent synthesis or through the advanced purification of existing materials. This means it's created with the utmost care to be as pure as technically achievable. Verification of authenticity. To confirm its authenticity, the reference standard undergoes an extensive series of analytical tests. These tests are conducted to ensure that the reference standard is an accurate and reliable measure for comparison, free from impurities and fully representative of the ideal form of drug. What is the purpose of reference standard? In pharmaceutical testing, the reference standard serves as the gold standard against which other samples or working standards are measured. Its high purity and rigorous verification process makes it crucial for maintaining quality control as it provides a consistent benchmark. Number 2, working standard. A working standard is a drug substance with verified quality and purity. It is established by comparing it to the reference standard, ensuring it meets required quality parameters for use in routine testing. Purpose of working standard. Unlike the reference standard, which is used less frequently due to its high preparation cost and purity, the working standard is used regularly in the laboratories. It serves as a practical tool for everyday quality control testing, allowing for consistent measurement and validation of the product quality. The working standard must match the reference standard closely in quality and purity. This comparison process involves analytical testing to confirm it meets the specification established by the reference standard. Next important term is an assay. What is an assay? Assay is a type of analysis used to determine the amount or concentration of a specific constituent in a mixture. In pharmaceuticals, it's commonly applied to measure the biological or pharmacological potency of a drug, ensuring that it has the intended effect. The primary purpose of an assay is to confirm the presence and quantity of active ingredients in a dosage form like a tablet or injection. For example, the assay of a vaccine is performed to confirm its potency, ensuring it is effective for its intended use. Assays are crucial in quality control as they verify that each product batch contains the correct amount of active ingredient. This helps in meeting safety, efficacy, and regulatory standards, ensuring consistency across all produced batches. Test. A test is a technical operation aimed at determining one or more characteristics or performance factors of a specific product, material, equipment, organism, process, or service. Each test follows a specific procedure to ensure consistent and reliable results. Tests are conducted to assess whether a product or material meets defined standards and requirements. In pharmaceuticals, tests are crucial for confirming quality, safety, and efficacy, making them a fundamental part of quality control process. The results of each test are typically recorded in document known as test certificate or test report. This document provides a formal record of a test outcomes which is essential for regulatory compliance and quality assurance. What is validation? Validation is the process of proving in alignment with good manufacturing practices that a procedure, process, equipment, material, activity, or system consistently achieves the expected results. It ensures that all the steps and components within a production system reliably perform as intended. The goal of validation is to establish documented evidence that provides a high level of assurance that a specific process will consistently yield products that meet their predetermined specifications and quality standards. This consistency is critical in industries like pharmaceuticals, where quality, safety, and efficacy are paramount. Validation serves as a cornerstone of quality assurance by confirming that every process from production to packaging reliably produces products that comply with regulatory and quality requirements. It reduces variability, minimizes risks, and supports regulatory compliance. Prospective validation, retrospective validation, and concurrent validation are the types of validation. Prospective validation refers to the validation process conducted before the implementation of a new product, process, or system. It aims to establish that the planned method will consistently lead to the desired quality outcomes when the process is fully operational. The primary goal of the prospective validation is to confirm the reliability and efficacy of a process before it is put into regular use. By validating the process in advance, manufacturers can identify potential issues and ensure that all quality specifications are achievable. Process of this validation, the prospective validation process typically involves several key steps: development of a validation plan, protocol design, execution of validation studies, data collection and analysis. Benefits: prospective validation offers several benefits and advantages. Ensures that processes are capable of consistently producing quality products from the outset. Reduce the risk of product failure and recalls by identifying issues before full-scale production begins. Helps in achieving regulatory compliance by demonstrating that thorough testing and validation processes are in place. Retrospective validation. Retrospective validation involves validating a process based on accumulated historical data and information for a product that is already in production and distribution. Instead of prospective studies, this validation method analyzes existing records to confirm that the process has consistently produced products meeting quality specifications over time. The main goal of retrospective validation is to ensure that established processes have been effective in maintaining product quality compliance with regulatory standards using data that has already been collected. The retrospective validation process typically follows these steps. Number one, data collection. Gather historical data and information from various sources including batch records, production logbooks, lot records, control charts, tests and inspection results, customer complaints or feedback, failed failure reports, service reports, audit reports. Data analysis. Review and analyze the collected data to assess the consistency and reliability of the process. This may include statistical analysis to identify trends, deviations, or areas for improvement. Evaluation of the results. Determine if the historical performance of the process aligns with predetermined quality standards and specification. And then the last, documentation. Prepare a validation report that summarizes the findings, conclusions, and any recommendation for process improvements if necessary. What are the benefits of retrospective validation? Retrospective validation offers several advantages. It utilizes existing data, which can be more efficient and cost effective than conducting new studies. It provides a comprehensive overview of the process performance over time, helping to identify patterns or recurring issues. It provides a comprehensive overview of process performance, helping to identify patterns and recurring issues. It supports regulatory compliance by demonstrating that established processes have consistently met quality requirements. Concurrent validation. Concurrent validation is a type of validation that occurs during the actual production of a product. It is considered a subset of prospective validation, as it aims to validate the process while simultaneously producing products intended for distribution. The primary goal of concurrent validation is to ensure that products being manufactured meet predetermined specification and quality attributes while the production process is ongoing. This validation approach allows for immediate feedback and adjustments if issues arise during the procedures. The concurrent validation process typically involves the following steps. Number one, real-time monitoring. Number two, data collection. Number three, evaluation of the results. Number four, review and approval. Real-time monitoring. Products are continuously assessed using non-destructive testing methods, which allow for verification without damaging the product. This could include various analytical tests and inspection to ensure compliance with quality standards. Data collection. Collect data on quality of the products being produced, documenting all results and observations during the validation study. Evaluation of the results. Analyze the collected data in real time to determine whether the process consistently meets quality specification. This may include comparing results against established control charts and benchmarks. Number four, review and approval. If the concurrent validation is being used for a new modified process, it is critical that the products are withheld from distribution until all validation data and result thoroughly reviewed. Only after confirming the process is adequately validated should the products be released. What are the benefits of concurrent validation? The benefits of concurrent validation are it allows for immediate identification and correction of issues during the manufacturing process, reducing the risk of defective product. It supports the efficient use of resources as validation occurs alongside production rather than as a separate phase. By ensuring that product meets quality standards in real time, it enhances regulatory compliance and customer trusts. Thanks for joining us in today's exploration of validation in the pharmaceutical industry. We hope you now have a clearer understanding of these definitions and prospective, retrospective and concurrent validation and their vital roles in ensuring product quality and safety. If you found this video helpful, don't forget to give it thumbs up and share it with your colleagues or anyone interested in pharmaceuticals. We'd love to hear your thoughts, so leave us a comment below. What topics would you like to cover? And remember to subscribe to our channel for more insights into the world of pharmaceuticals and quality assurance. Until next time, keep striving for excellence in your work and stay curious. Thank you.